Hello students, I am Sharnuporni and I am from Learned Education, and I am here to take you through the absorption by roots and rise of water up to xylem. Now this is the fourth chapter in our class 10 ICSE syllabus. This is the new syllabus, revised one, published by the ICSE board in the month of November. So with this chapter we are going to start off with the plant physiology, and as you can see, first of all we can divide the chapter into two stages. One is the absorption of water and minerals by the roots, and next, how does this water is getting transported through the xylem. I'm going to take you through the next slide, which is absorption by roots. Now what do the plants take up in their roots from the soil? They take up two things. One is water and the other one is minerals. Now water is needed for what? It is needed for transportation. Transportation of what? Transportation of food, transportation of minerals. Now I'll be giving you short forms of this. You have to understand food, minerals and of other ions and solutes which are getting transported throughout the plant body. Next is the transpiration. Now transpiration of water is a very important phenomenon. You might have learnt in your previous classes that transpiration is the evaporation of excess water from the aerial parts of the plant. So that is being taken care of by the water. Because the water is there, in order to balance that water in the plant body, transpiration takes place. And also transpiration is helpful for the ascent of sap. As you will learn in the future class, in the future slides, that transpiration helps in the ascent of sap, that water is being taken in by the plant because some water is getting evaporated. Water is being taken up by the leaves of the plants. Okay, in this way water is getting transported. In this case, in this way ascent of sap is taking place. Photosynthesis is the third one. Photosynthesis, we need water for photosynthesis. It is one of the raw materials for photosynthesis in order to make food. Then water provides turgidity to the cell. What is turgidity? Turgidity is the distension of the cells. Cells get distended because of the presence of water in them. So turgidity, what is the function? Turgidity provides mechanical support to the plant body. Turgidity helps in the movement of the plant. In movement means stomatal pore opening and closing, in the germination of seeds, and also like in the opening of flower, the flower blossoms because of this turgid pressure. We are going to talk about it elaborately in our next slides. And lastly, the germination of seeds. Now as the seeds take in water, they burst, the seed coat bursts and the new plantling comes out. You might have learnt also about the minerals part. Why do the plants need minerals? Now minerals are needed by the plants because they are constituents of cells and cell organelles. So these minerals like iron, like nitrogen, like sodium, like potassium, molybdenum, all these minerals are taken in by the plants. You can further divide the minerals into micro and macro elements. And lastly, synthesis of various enzymes and chemical compounds which is required because of the presence of the minerals. For the presence of minerals, these things can be made. So that is why we need water, we need minerals, and how the plants are getting these water and minerals? By the roots. Now we are going to be studying elaborately about this absorption process. Now this is one topic that is given in your syllabus. If you note that understanding of the processes related to absorption of water by the roots, and the first process in it is imbibation. What is imbibation? It is the absorption of water molecules on the surface of colloidal substances, colloidal or solid substances in living or dead cells. Right. So, what do you understand by that? Now, imbibation is a type of diffusion in which what is happening? In which the plants are absorbing, the root cells are absorbing water. Cells of the roots, cells of the stems, any cell living or dead, they are absorbing water, and they are, as a result of it, they are increasing. I am writing increasing short form INC, increasing volume. Now, imbibation is a process which you can see in your day-to-day activities, like you often see warping or the swelling of wooden doors and windows. So in the rainy season, what do we see? We see that there is a problem in locking the door because the hinges, they swell up, because the wood that is being made, that is used to make up the doors, that is absorbing the water and it is swelling up. So that is one phenomenon we can observe as a result of imbibition. Then there is another one, which is the swelling of seeds. Now the seeds, they take up the water and they swell in size, they are full of water, they absorb the water. Lastly, the significance of imbibition pressure. Now what are the after effects? After the seeds imbibe water, they germinate. So germination of seeds, the seed coat bursts open and the plantling comes out. Uptake of soil water by root hairs, the minute unicellular root hairs. We will talk about the anatomy of roots in our next class. But for today, you must know that the uptake of water that is present in the soil is happening because of the root hairs, and that uptake is known as imbibition. Movement of water from cell to cell. As one cell gets filled up, that water is transferred, a portion of it is transferred to the next cell. So this cell to cell imbibition, this transfer of water from one cell to another is happening because of imbibition. So we will learn a bit more about it. This is an experiment you can perform even at your home. What you need to do is, you will take a can and you fill it up with dried gram seeds. We all have dried gram seeds in our kitchens. So, and then you add water to it, you fix the lid and keep it for observation. Like for a night, you observe it, and in the next night you will see if you have added enough gram seeds, like you can see in the picture. The lid bursts open. The lid bursts open and all the swell seeds, they are coming out, they are tumbling out of the can. Why does it happen? Because the dry seeds, I am writing out here, the dry gram seeds have taken up, have imbibed water and swelled. Imbibed water and swelled. This is the first phenomenon of processes that is happening in the plant to absorb water. So definition, the definition of imbibition can come for your exam as a 2-mark question, and also they can show you a picture of this, or they can ask of you about the experiment which you can perform to show imbibition in seeds. Next slide, it talks about the diffusion process. Now, how is diffusion happening? We all know, we have learnt it from smaller classes, younger classes, that diffusion is the free movement of molecules from region of its high concentration to its low concentration. That means, suppose I am spraying a bottle of perfume in this corner of the room, the person who is standing at a far corner of the room will be able to get that fragrance. Why? Because the fragrant molecules will through the atmosphere and will reach another space, another empty space. So now this also have the molecules here. Okay. So this is diffusion. Diffusion can happen in solutes, in liquids and in gases. So you can very well write substance in for substance you can write diffusion is happening in solutes, liquids and gases. You can also give the example of dissolving of sugar in a cup of water. So we cannot see it, but you can obviously taste and understand that if you take a beaker of water, add sugar molecules to it, without stirring, if you keep it like that, sometime later the sugar molecules will dissolve by itself and all the molecules will be dispersed and diffused throughout in between the molecules of water. So that is dissolving of sugar in a cup of water. Significance of diffusion. Now, just like imbibition, significance of diffusion is also very important. These can come up as 2 marks direct question, so you better pay attention to it. Gaseous exchange between atmosphere and leaves. So as you imagine a plant, imagine a tree, and you can, this is the foliage of the tree, these are the leaves. So in these leaves, if you go to the leaf level and if you understand it, what would you understand? That carbon dioxide is being given out, oxygen is being taken in. All these is happening through the stomatal pores, and this is because of diffusion of these gases. The movement of solutes and solvents between adjacent cells. So as we spoke about just now, that imbibition is taking up of the water, and the transfer of water through the adjacent cells is taking place by diffusion. And also loss of excess water from leaf surface. This process is again, it is known as transpiration. So transpiration, from one cell water is moving and finally it is being transpired to the environment. That is also happening as a result of diffusion. Now we come to this particular picture which explains the process of diffusion. What happens here? Now in this case we can actually observe, we can see the change. Here we have immersed one permanganate, potassium permanganate crystal, which is reddish pink in color, in a beaker containing clean water, clear water. After some time you can see that this entire clear water is slowly becoming reddish pink in color. Why? Because this permanganate crystal is diffusing into the water molecules, through the water molecules, and as the diffusion is completed you can see the color change. So this proves that diffusion can take place, and diffusion can happen in solutes, solvents and gases. In the third process we talk about osmosis. We will talk about the difference between the two, diffusion and osmosis, but first, what is osmosis? Osmosis is the movement of solvent molecules. Note this down, solvent. Okay, let me use a different color. So this is the solvent molecule. What kind of solvent molecules? All the liquid molecules. They, from its higher concentration, from molecules higher concentration to its lower concentration through a semi-permeable membrane. Okay, semi-permeable membrane, you can give many examples like parchment paper, like cellophane paper, and if you're talking about living cells, you can say the plasma membrane or the cell membrane, semi-permeable membrane. Now, though we have semi-permeable here, it would be appropriate if you use the word selectively permeable. Selectively permeable membrane in process of osmosis. One is endosmosis, another one is exosmosis. The difference between the two you must note because this comes up in your exams. What is when there is a shrunken cell in the medium and the medium contains a lot of water. Suppose we are talking about a resin. You know resin, we often have raisins are very sweet. Now raisin, if you keep raisin in a beaker of water, you will see, or a few raisins in a beaker of water, you will see that the raisins after a point of time they have. So in that case what happens, the water molecules have entered these resin through the membrane of the raisin. Therefore it is endosmosis, where water is entering into the cell. Exosmosis, if you keep very swelled up raisins in suppose a sugar solution, a concentrated sugar solution or concentrated salt solution, what you will observe, the swelled up raisins, here I am giving a more bulging raisin for an example. If you keep it in concentrated salt solution, you will see after a point of time the raisin cells, the raisins have shrunken, where water has moved out into the beaker, into the medium where there was concentrated salt solution. So what basically happens? This is an example of exosmosis. So what is happening in this picture? We are saying movement of water molecules through semi-permeable membrane. So there is a membrane. Water molecules are high in concentration towards the left side and low in concentration towards the right side. There are sugar, these big brown molecules are the sugar molecules. So water, which is the solvent, is going to move from region of high to region of low concentration. The arrows suggest that through a selectively or semi-permeable membrane. And what is endosmosis? when the cells are taking up water, when the water is moving inside, this is exosmosis, when the water is going out. So depending on the medium on which you are keeping it, this phenomenon will happen. That is osmosis. Now osmosis is basically happening everywhere, in our cells, in the plant cells. Now what would you say absorption of water by the plants as a significance? So as water is being taken up, osmosis is happening from cell to cell, and even from the soil to the root cell, that the water is being taken up, that is happening by absorption, by osmosis. Then allows movement of water from cell to cell, one cell to other cell. From, suppose there are two adjacent cells. So what will happen, this is a cell which has higher water concentration, solvent concentration. Cell has lower water concentration. So I am putting a cross here to show lower water concentration. So water molecules will move and towards this side. So this is from cell to cell, and also opening and closing of stomata. As we will study further, we will know that how this osmosis is helping in the opening and closing of stomata. That is, when the water is entering the stomatal cells, the guard cells, how this process is happening. So that we will see in later slides. Next we move on to the next slide, which is demonstrating the process of osmosis. This is a laboratory demonstration. What they have done, they have taken a thistle funnel, they have filled it up with 25% sugar solution, and they have immersed this thistle funnel in a beaker containing water. What do? You fix it up with and note the initial level of sugar solution. Initial level of sugar solution has been noted, and the initial water level, which is high up, after a point of, okay, I have spoken about the thistle funnel filled up with sugar solution. Here in the base of the cellophane paper. Now as I said earlier, cellophane paper is a semi-permeable membrane. So as we immerse it in a beaker containing water, observe after a point of time that the level of sugar solution inside this thistle funnel has increased, and water level inside the beaker has gone down. So if you notice the final level of sugar solution, it is high up in the thistle funnel, higher up, and the initial water level and the final water level, if you notice, the final water level is further down than the initial water level. So you will be able to say, tell me what is happening here. The water molecules are entering the thistle funnel. So inside the thistle funnel, more concentration of solute is there, less concentration of water. Outside the thistle funnel, more concentration of water. In the beaker, more concentration of water is there. So water is entering. Water is entering. Water molecules being the solvent molecules are entering into the thistle funnel and rising up the level in the thistle funnel. This is what is happening in this experiment. Now in the second experiment, for your better understanding, this is also a very important diagram, often comes up in your exams, in the particularly in the second section, where you have to there are 4 questions you have to attend, 10 marks each, and there are part markings. In those part markings, they give you these kinds of diagrams. They ask you to tell them how these phenomenon is taking place, where water is moving, in which direction, or in which of these cases water level will rise. We will see. So it's a beaker. You have taken potatoes. First you take 2 potatoes and you flatten their base. You scoop out the starch inside. You peel off the potato covering, the brown covering, you peel it off. You scoop out from inside so that you can have this cavity. Flatten the base so that these potatoes can be placed in the beaker containing water. In one of the cavities, you fill in that 25% sugar solution. You fill the beaker with water as well, and you fix a pin to notify the level of sugar solution. Okay, the pin is here. That is the initial level of sugar solution inside the potato. And in the second, this is the control experiment where the experimental material is missing. So in this control experiment, you have the water in the beaker and you have this potato, and you have water inside the potato cavity. Here you have sugar, here you have potato. Now this setup is known as potato osmoscope or potato osmometer. Any language can come for your exam. So what do you observe after a period of time? You again, because of osmosis, water molecules will move inside the potato because it has lower concentration of water, and from higher concentration of water, that is in the beaker, to lower concentration it will move and the sugar solution level will rise. Because of that pin you can easily observe it, but in this case there is no rise in the sugar in the water level inside the potato cavity. Why there is no rise? Because here it is water, here it is water. So there is equal concentration in outside and inside of the potato. So no movement of water is taking place. So in this case we talk about there is no movement. In this case there is movement. So osmosis, where is it happening, children? It is happening in case of A. Okay. Now move to the next slide. Now this is not a process of absorption. These are two terms which are very important for understanding, which is a result of these processes. One is osmotic pressure. I will explain it through this experiment. First let me tell you the definition of it. It is the minimum pressure that prevents the passage of pure solvent into the solution when they are separated by a semi-permeable membrane. So if you go back to we go back to this slide where water was entering into the thistle funnel, what do you think, for how long this entering of water will happen? At some point this osmosis process will stop. When will it stop? When the concentration of both will be equal? No. Before even the concentration gets equal, the osmosis process will stop. When the water column inside the thistle funnel will start exerting a pressure on the water of the beaker that is entering the thistle funnel. So if you go back to the slide, this is the slide which is, this is a picture describing the osmotic pressure. So in this picture you can see that the setup is the same. Cellophane paper here acting as semi-permeable membrane. The thistle funnel is here. Water molecule, the sugar solution, 25% sugar solution has been filled up in the thistle funnel. Water is here. You note the initial water level, you note the final water level, note the initial sugar level solution, and after the experiment we will note it, the final sugar level solution. We also put a piston inside the thistle funnel. Okay, we put a piston inside the thistle funnel. Now what will happen as osmosis will start, water molecules will start entering this thistle funnel. Now as it enters, we will do one thing, we will put weight on the pistons. Keep on putting weight. Now when this weight will exceed the weight of the pressure of the water molecules that are entering this thistle funnel, when this pressure, this is a weight which is pressure, this pressure is exceeding the pressure of the water molecules entering the thistle funnel, then osmosis will stop. So that pressure which we need for osmosis to stop is known as the osmotic pressure. And this can come up in your definition section where they can ask you about the definition. They can of course ask you about to describe or to tell you the observations in this case, in this experiment. What is your observation? That after a point of time osmosis will stop because the osmotic pressure is there, because it has reached the osmotic pressure. Then osmotic pressure gives rise to root pressure also. It becomes root pressure when we are talking about plant root system. What happens? Pressure generated in the roots due to continuous inward movement of water through cell to cell osmosis. So now if our material, if we are talking about, let me use a black ink here. So now we are talking about these cells. Suppose these are all plant cells which are placed adjacent to each other. And what kind of cells are these? These are the root cortical cells. We will talk about the anatomy of the root section in the later class. In today's class, let us assume that these cubical cells are the root cells. So this is the soil particles, and water from the soil particles is entering these root cells first. So as the water is getting accumulated, there is a generation of root pressure, the pressure inside the cell. This root pressure will then allow the water molecules to transfer from this plant cell to the next plant cells, to the adjacent plant cells. The root pressure, basically the application of root pressure is the transfer of water molecules through the adjacent cells of the root. Okay. So, application is important and definition is also important in these two cases. And then we talk about that age old question, that what is the difference between diffusion and osmosis. Now, when you are writing diffusion and osmosis, when you are writing differences between the two, I prefer, or it is preferred, the examiners prefer it to see it in a way that you present it very nicely. Suppose this is diffusion and I am putting a line in between, and this is osmosis. Write a parameter, write parameters over on the left side, like on which parameter you are judging it. So, if I write the parameter to be, if I write it like what is diffusion, the definition, the parameter is definition. So, according definition, diffusion is the movement of solute, solvent or solid, liquid, gaseous molecules from region of high to low concentration. Osmosis is the movement of liquid solvent molecules from region of high to low concentration across a semi-permeable membrane. Very important definition. So, this you give in for definition. Then semi-permeable membrane, the next parameter is semi-permeable membrane. Presence or absence of it you have to determine. So, this is the parameter you are writing on the left hand side. On the right hand side, for diffusion you are writing no semi-permeable membrane is required. Of course, you do not give a cross, you write it in words, and osmosis you give a tick. That means semi-permeable membrane is there. Then for diffusion, what else can you write? Diffusion, as it can happen in gases, also a much faster process. Now, if you write the, suppose you write the rate of reaction, rate of the process, then diffusion will be much faster. So, it is faster or rapid, whereas osmosis will be rather slow or slower than diffusion. Lastly, you can write the substances which are getting diffused or osmosed. So, for the substance you can write diffusion can happen in solutes, in liquids and in gases. Now, my writing may not be appearing very clear, but I hope you are side by side noting what I am saying. Solute, liquid and gases, whereas for the case of osmosis, it is only solvent molecules are getting osmosed across the medium. So, that is the difference. The difference between these two can come up in your 2 marks question. It is preferred to write the parameters in the left hand side and then write whether it is absent or present in most cases where you can do it, for the examiner to understand in a better way. Now, there is another phenomenon which is not exactly a process, but it is a phenomenon that is happening. We need to talk about it since we are talking about osmosis and diffusion. The phenomenon is known as tonicity. What is tonicity? Tonicity is the relative concentration. Tonicity is the relative concentration of two solutions that determines the direction of movement of the solvent. Now, in these cases, what you will assume is, we will assume a plant cell. Plant cells are somewhat rectangular in shape, immersed in or kept in a medium. Now, what is there in the medium, or what is the constituent of the cell sap, we will discuss as and when we will proceed. So, this is the plant cell, the cell sap of the plant cell immersed in some medium. Okay. Now, first is tonicity is the relative concentration. Now, it has some types. One is isotonic solution. Isotonic solution, where iso means same and tonus means tension or concentration of the solute. When the solute concentration in the cell and in the medium in which it is immersed both are the same, then it is known as an isotonic solution. That means the water will neither move in nor move out. This won't happen. No osmosis is taking place in here. Why? Because concentration of both these are the same. In case of hypotonic solution, if we talk, hypo means low, lower concentration of solutes and higher concentration of solvent. There is an arrow missing, which is end osmosis. So, if we have to erase this part. Suppose we are erasing it, we will draw it again. This is the second condition. So, in the second condition we have the cell again. Lower concentration of solute in what? In the medium. The medium has lower concentration of solute, more water is there, more solvent particles are there. More solvent are there. So, what will happen? And less solute is there, and here more solute is there and less solvent is there. I hope it is clear. More or less you have to know the medium is very watery, less solute, more watery. So, you know that what will happen from the region of its high concentration, water will move into the cell and end osmosis will take place. So, hypotonic solution, you put a cell in a hypotonic solution, in a solution containing less number of solute, more solvent molecules, the cell will take up the water in osmosis. And in contradiction to that, hypertonic solution where hyper means high concentration of solute. Now, here we write high concentration of solute, we will just flip this. We will flip it. Here it will be less solvent, more solute. Wait, let me erase this part also. Okay, here it will be in hypotonic solution, higher concentration of solute and lower concentration of solvent leading to exosmosis. So, here there will be less solvent and more solute, okay. And inside the cell sap has a lot of water, less solute and more solvent, okay. Hyper means high of what? High concentration of solute. So, then what will happen? The cell will lose water and water will move outside of the cell, a phenomenon you already know, which is exosmosis. So, this is a very important part of this chapter, where they will ask you definitely where, in which direction water is moving. Whether if you have to judge whether you are keeping this setup, the cell, your setup or specimen in a hypotonic solution, means containing less of solute. So, in that case what will happen? The cell will take up water. Or in a hypertonic solution, containing more of solute, the medium contains more solute. So, in that case what will happen? Cell will lose water. And if the concentration of the medium and the cell are the same, then there is no movement of water, that is an isotonic solution. So, this is your concept. We have almost reached towards the end of the class. Okay. So, here we have turgidity. The next phenomenon, the phenomenon of turgidity is here. So, if you see, when the cell vacuole of water pushing the cell cytoplasm against the cell wall, allowing no more water to enter, the condition is called turgidity, turgid cell, and the cell is said to be turgid. So, what is basically happening? Now, we have spoken, until now we have spoken, cell is taking up water, cell is taking up water. So, what is that thing called when the cell is completely distended, swelled by taking up water? So, this is the cell, and here the cell, this is the vacuole, if you assume, this is the vacuole, this is the cell wall. So, this is the cell wall, I am depicting it as CW, and this is the cell vacuole. Cell vacuole, the water is getting accumulated inside the cell vacuole. Cell vacuole is gradually enlarging and when it is totally turgid, when it is enlarged to its highest level, that is known as turgid. The cell is then said to be as turgid cell. It cannot swell in anymore. Okay? So, that gives rise to a pressure known as Turgor pressure. Again, I will use a different pen color for you to understand in a better way. So, Turgor pressure. What is Turgor pressure? When cellular contents are putting a pressure on the cell wall, okay, because of turgidity, That is known as Turgor pressure. Very important terminology, often asked in your exams. Turgor pressure is the pressure which is exerted by the cellular components on the wall of the cell as the cell becomes fully turgid. As you know, must have read in physics that to every reaction there is an equal, to every action there is an equal and opposite reaction, Newton's third law. So according to that third law, then the cell wall will also exert certain pressure onto the cellular content, and that is known as wall pressure. The cell wall is pressurizing, in this direction you have to picturize. The Turgor pressure was towards the cell wall and the wall pressure is towards the cellular content. So this is known as wall pressure. Okay. So this is the significance of, this is the definitions of turgidity. Then flaccidity. What is flaccidity? Flaccidity is the loss of water resulting in shrinkage of cell contents. As the cells are taking up water, they are becoming turgid, and when the cells will lose water, suppose a lot of evaporation, lot of transpiration is happening in a hot sunny day, the plant cells will give out the water and they will turn flaccid, they will wilt, they will crumple down, they will be looking dry. So that condition is known as flaccidity, loss of water due to shrinkage in cellular contents. In the next section of the slide, we are going to talk about the significance of turgidity. Very important phenomenon, has a lot of significance. It brings about movement of water and thus generates root pressure. So turgidity, root pressure, that is happening because of osmosis. So osmosis is leading to, let me use a different color, leading to root pressure, and root pressure is leading to Turgor pressure or making the cell turgid. Okay. So as this cell is getting, one cell is getting turgid, it will readily lose the water and it will transfer the water to the next cell. So in this way, cell to cell osmosis will take place. So turgidity is actually brings about the movement of water and it also generates root pressure. So it is a continuous cycle of phenomenon. Again, turgidity is helping in osmosis, osmosis is creating the root pressure. This cycle is happening as the water is moving from one cell to another in the roots. Essential for growth. How is it essential for growth? Because you can see that if I draw a small cell out here, as the cells will grow, obviously they will divide, but they will take up water and they will swell up. So the cell wall gradually gets extended in a process which is resulting in growth. So turgidity is giving rise to a distension of the cells and hence growth of the cell. Then what happens? Maintenance of shape, maintains shapes of the cells and gives rigidity to the soft tissues. So as we talk about young plantlings, small seedlings, we talk about there are lots of parenchymatous cells, lots of dividing cells, meristematic cells. Those cells need the rigidity that is provided by Turgor pressure, by turgidity. As the cells are taking up water, they are becoming more rigid. That turgidity is giving mechanical strength to the cells. And lastly, this got hidden by this picture. It says that it regulates cellular movement. I am writing it down over here. Turgidity regulates cellular, wait, I will erase this part, root pressure, because it will confuse you. We have already covered it, so I am erasing it. So turgidity is regulating cellular movement. How is it regulated? Remember, in the previous slide we have studied how osmosis is helping in opening and closing of stomata. This is what it is. What is happening here? Because of osmosis, because of the transfer of cells, the water is moving into the guard cells of the stomata. You know the stomata is there. It is an opening through which gaseous exchange take place, and stomata is helped by these guard, it's flanked by the guard cell. The stomatal pore through which the gaseous exchange is taking place is flanked by the guard cells. Now as water is entering into the guard cell through the cell to cell osmosis, the guard cells are getting swelled up. It is becoming turgid. Epidermal cells are next to the guard cells, so it is getting turgid. So because of that, the cellular pore, the stomatal pore, that is opening, that is opened up, okay. The guard cells are taking away the stomatal cells from the stomatal pore. Stomatal pore is opening. And next what happens, as flaccidity happens, as the cells will lose water, because the water gets evaporated out of the stomatal pore, then the guard cells, they turn flaccid, they lose water and they become flaccid at night. And the stomatal pore, they close. Flaccid, close, and turgid, open. Remember this, turgid, open, flaccid, close. Now this is a very important picture. They can ask you to identify the phenomenon, of course. They can ask you to label all these parts of the stomata, which phenomenon is happening, discuss the phenomenon, describe the phenomenon, and also what is the function of the guard cell. Guard cells are guarding the stomatal pore, just guarding and helping, aiding in the opening and closing. Next, regulate cellular movement. We have the example of Turgor movement in the leaves of touch-me-not flower. So, I hope you have seen a touch-me-not flower, and its scientific name is Mimosa pudica. What happens that in normal condition, the cells are completely turgid, okay, they are swelled up, the cells of the leaves are swelled up. But if you touch it, if you put your finger in between these cells, so this is the mid vein. Now there are number of leaves here, okay. So if you touch somewhere here, what will happen? The leaves will droop. The leaves will droop down, and this is as if the plant is getting shy on your touch. It is not, it is what is happening is the plant is losing the water, the cells are losing the water and the cells are turning flaccid and therefore drooping off. Again, when you put away your hand, they will slowly gain the water and they will form turgid. Now one thing I need to mention out here is in the cells, if you can see, these are the leaflets along the mid vein. So in these leaflets, if you look closely at the base of the leaf, you will see a solen base. This solen base is known as, let me write it over here, it is known as pulvinus. This pulvinus is actually guiding the intake of water and the losing of water. So turgidity and flaccidity is happening through the pulvinus of the touch-me-not flower, which is at the base, the solen base of the leaflets is known as pulvinus. So this is the process of turgidity. I hope it is clear. When you talk about the definition, of course, then you slowly move on to the important terms, Turgor pressure, wall pressure, very important. Flaccidity, how the water is lost, and significance, number of significance of turgidity. Amongst this, regulation of cellular movement is most important, because this has come up in your previous year's examinations also, where they give you this diagram and for you to identify. Okay. Then we come to this phenomenon known as plasmolysis. Now what is plasmolysis? This is also a phenomenon which is related to the absorption of water and the relatable phenomenon. It is the shrinkage of protoplasm due to loss of water when kept in hypertonic solution. Deplasmolysis, opposite of plasmolysis, is protoplasm back to its original position on gain of water. So first we will learn about the process, then we will go to its practical applications. So if you see this is a normal cell, or you can call it as, it is a turgid cell, where there is enough water, everything is fine, this is a normal cell. Now as you put this cell in a hypertonic solution, means in a solution containing more of solute. what will happen? The cell will lose the water, the excess water it will lose, or excess or more than excess water it will lose to give to the solution. So, osmosis will happen from the cell to the medium, that is exosmosis, outside of the cell is happening. So, cell will gradually show contraction of the cytoplasm. So, cytoplasm, if you can see, it is, or protoplasm, it is getting constricted away from the cell wall. Away from the cell wall, away from the cell wall, and finally it is becoming a plasmolysed cell. A cell membrane and the cell itself, cytoplasm, has constricted and it has got into around the middle or around other in a corner of the cell it will go. And the cell wall is there. This content, this is not the cytoplasm, this is the empty portion of the cell. So, this is now known as a plasmolysed cell, where the entire cellular content is constricted to one small portion. So, because of the loss of water. Why the loss of water? Because the cell has been kept in a hypertonic solution. Now, what happens to deplasmolysis? What happens when deplasmolysis takes place? If you put this cell, if you take it up from the hypertonic solution and you place it in a normal cellular medium, again in a hypotonic solution where there is more of water, less of solute, again then what will happen? Endosmosis will take place. Water will enter from the medium to the cell. So, at that time it will gradually revert back into its original position. So, this original position reverting back to it, it will finally become the deplasmolysed cell. Deplasmolysed cell, where the cell membrane is again getting near the cell wall, and it is a normal cell you will get. So, plasmolysis when you keep it in hypertonic solution, deplasmolysis when you keep it in normal hypotonic, in a normal solution or a hypotonic solution. Okay, so this is what it is. This is your this diagram is very important for your further questions. Further questions can be asked. This has been asked in your previous classes also. We will move on to our previous slide only. We have omitted this portion, practical applications of plasmolysis. Now, how can you apply plasmolysis in day-to-day basis? Now, you might have seen that salting of meat or pickles are done to keep them preserved for longer time. Why does that happen? Why do that? We do that to kill the bacteria or the germs. So, basically when you plasmolyze the bacterial cells, you add salt, you plasmolyze the cells, and you prevent the meat, the vegetables, the pickles from getting rotten. You can we often do this killing of weeds, suppose in our playgrounds or in places where we do not want these weeds to grow, where we are maintaining a garden. What can we do? We can add salt, a lot of salt to the roots of the weeds. So, the cells of the root will be plasmolyzed. So, killing of weeds by applying salt, cells. Root cells as a result will be plasmolyzed. This is your final result. Application and the application of plasmolysis, where you can see plasmolysis and deplasmolysis. Now, this is the last part of our today's lesson. After that, we will delve into questions. We will study the questions, probable questions that will come up. Active transport is the last part. What is it? It is a movement of substance from its lower to higher concentration with the help of energy. Now, this seems to be just like any other definition we are learning from the start. No, it is not. If you notice, this is from lower to higher. This is not from high to low. This is against the concentration gradient. So, I am putting a bracket out here, against concentration gradient. So, active transport takes place against the concentration gradient with the help of energy. Transport happen? It takes place only to transport minerals and ions, no solvent molecules. ions and minerals are getting transported. So if you have, suppose you have soil in this beaker and you have one cell, I am talking about one cell, this is a hypothetical situation, but I am actually talking about when you are planting a small plant and there are root cells there. So how will they take up the minerals? We have spoken about how they will take up water. We now will know how will they take up minerals. So the minerals will be taken up from their soil against the concentration gradient into the cells of the root, and or the ions that can move high concentration, and minerals in the soils are more, but they are also moving with the help of active transport. Talk about against the concentration gradient, that will also happen because of active transport. Okay, so in the membrane, across the membrane it will happen, and in the membrane you have certain proteins which are known as carrier proteins. So you need a semi-permeable or selectively permeable or living membrane for active transport to happen, and also you need membrane proteins. I am writing all the important terminologies down that needs to be included in the broader definition. Membrane proteins are embedded in the membrane which are taking up these molecules inside from low to high against the concentration gradient with the help of ATP. The minerals also get transported with the help of active transport, with the help of ATP, okay, when the plant take up the minerals from the soil. And passive transport is the movement of ions across cell membrane from the higher concentration to the lower concentration without the need of energy. So can you tell me what is the difference between osmosis and passive transport? The difference is ions is transported, and in osmosis only solvent molecules, and osmosis is actually a type of passive transport. Passive is more encompassing, it is transporting ions, molecules and everything, solvent molecules, whereas osmosis is only transporting solvent molecules. So osmosis is a type of passive transport. Now we talk about the difference between diffusion and active transport. Again, we will make a chart, we will make two columns, actually three. One will have diffusion in it, one will have active transport. Wait, let me write it nicely. This is active transport in this side, and here is the diffusion, and here is the parameter on which we are differentiating the two. This is very important, these questions come up as it is. So parameters, what do we have to say? We have to say the definitions first. So in definition we will say that diffusion is the movement of substances from region of high to low concentration, and in case of active transport we will say that is, it is the movement of substance from its low to high concentration with the help of energy across a semi-permeable membrane. You can add that in the definition part, or you can make up other parameters also. So if you write semi-permeable membrane here, what you will say that diffusion needs or doesn't need. It does not need a semi-permeable membrane. Again I am putting a cross, you will write it in your words. Active transport, it needs a semi-permeable membrane. If you are talking about membrane, you must talk about the membrane proteins. Diffusion is taking place without help of any membrane protein. It doesn't need membrane, it doesn't need the membrane protein. It needs the membrane, it needs the membrane protein. Fourth point can be diffusion, whether energy is required or not. Energy requirement. This I am just striking it off. Energy requirement is not there in diffusion. External energy is required in case of active transport. Why? Because diffusion, the transfer is happening along the concentration gradient. The gradient itself is facilitating this movement. But in case of active transport, external energy is required because it is happening from low to high. And lastly, you can say that diffusion can happen in solid, liquid, gas, whereas active transport is only happening in case of solute molecules. Okay, if you can think of any other difference between the two, then obviously you are permitted to write that. Whereas I can think of these only, these almost make up the definitions of these two processes as well. Okay. So these are the entire thing. So what all have we studied till now? If we see it, if you have a quick recapitulation, we first spoke of the absorption of by roots, what all the roots are absorbing, water and minerals. Water is needed for which processes, minerals are needed for which processes. Then we spoke about the understanding imbibation or understanding of the processes. Under that we studied the first process, that is imbibation. You can quickly go through the definitions and then the experiment to describe imbibation. Then we spoke of diffusion, how movement of molecules is happening from high to low concentration gradient, and examples of diffusion we have studied, significance of it. Then diffusion experiment to prove diffusion. Then we have osmosis. It is one of the most important processes, which is the term comes up in every other chapter that we will study, cell to cell osmosis. What is that? We can see in the slide. Then the osmosis, it's a laboratory setup. We can study studying osmosis, we study osmotic pressure, which is stopping the process of osmosis after a period of time. And osmotic pressure gives rise to root pressure. Difference between diffusion and osmosis. Tonicity, it is one important term that is important to understand which mediums do we use, which kind of medium. So how we are talking about the transportation from which to which cell, from a less concentration cell to a high concentration cell or vice versa. Those understandings come from tonicity, whether it's an isotonic or a hypotonic or a hypertonic solution. Then we spoke about turgidity of plants. When will the plant get turgid? So turgor pressure, wall pressure difference comes very often. The difference of it and the significance. Plasmolysis, almost one of the last ones, where we spoke about the shrinkage of protoplasm away from the cell wall. And lastly, we spoke about the active transport. Okay, so the parameters say definition, semi-permeability of the membrane protein, energy is required in the difference between diffusion and active. You can add one more point, is the concentration gradient. This is happening along the concentration gradient, this is happening away from the concentration gradient. Passive transport, it is relatable to the ions and it does not need energy. Okay, now we come to the questions part. Application skill based question. These normally come up in the second part of your question paper, where they give you 10 marks question. Part marking will be there. So it's very important for you to go through the question, to go through the diagram in order to understand the question. Diagram is of a setup is a very important physiological process has been given. You have to name the process. So what process is happening? The process of osmosis, isn't it? Explain the process. You can very well explain the movement of solvent molecules from region of high concentration to low concentration across a semi-permeable membrane. That is the explanation. What change would you observe in the thistle funnel containing sugar solutions after 10 minutes? What will you observe? That the level of sugar solution has risen. Level of sugar solution has risen, has gone up. So that is your observation. I am telling you the answers, but you also have to understand these questions very properly. You have to make up the answers before I utter them out. So you think along with me. Is sugar solution hypertonic or hypotonic? Hypertonic means concentration of solute is more in the solution. That means sugar solution is hypertonic. That's why the hypo from this beaker which contains more solvent water molecules, water is entering. This one is hypotonic, the water in the beaker, this one is hypertonic. Name the part of the cell represented by the sugar solution. So if you, this won't be the sugar solution, this will be the cellophane paper which is present. This is the cellophane paper. The question is wrong. Oh no, wait a second. No, this will be sugar solution only. Let me delete it. Which part of the cell is represented by the sugar solution? Of course, if we take up this sessile funnel as the cell, the sugar solution is the cell sap. So the answer will be cell sap. Answer is cell sap. So these are the questions. Now next one. Again a diagram based question where they have given you two diagrams of plants. Now you have to determine a few of the things. Name the structure shown. Structure is stomatal pore. Okay. Structure is stomatal pore. Okay. Name the parts numbered from 1 to 5. Now we have spoken about turgidity and all that. So this is an eye opener for a question. We have to also know all the parts individually. So what are the parts? Number 1, what does it show? I am giving you 2 seconds of time to understand what it is. It is, these are small spheres inside a guard cell, okay. So this will be, one will be the chloroplast, the green chloroplasts that are there in the leaves. That's why we cannot see the stomata in naked eyes, which very minute microscopic pores are there, and they are also taking up the pigment chlorophyll. So this is the chloroplast. Okay, what is number 2? 2 is, if you can see, it is just these lips of the pore. So this will be stomatal pore, stomatal pore. The third one, these big spheres, these are the vacuoles which are taking up the water, making the guard cells turgid. So these are the vacuoles. The fourth one will be guard cell, of course. The stomatal pore flanked by the guard cells. Guard cells. Fifth one is just, they are showing this stomatal pore only, but they are putting that arrow towards in the middle. So this is the stoma or stomata itself. Stoma is the plural form, stoma. So all these have been identified. Number 1, number 2, 3, 4, 5. Okay. What is the difference between A and B? The cell A is more turgid, where the stomatal pore is open. Cell B is flaccid, stomatal pore is closed. Name the mechanism which brings about the change in the structure of A, turgidity and flaccidity. Discuss the mechanism in brief. Here you have to discuss how from cell to cell osmosis, cells are gaining water and they are becoming turgid, opening the pore, and flaccid, closing the pore. In the next question we have these two diagrams. All are mostly I have given diagram based questions because from this chapter this is very important. Short definitions, differences, answer the following, give reasons can come for sure, but these diagram questions are difficult to understand if you are not very well adapted to the chapter. So you see these two cells under the microscope which are placed into different solutions. You have to say what is the technical term for the condition of cell A. This is a normal cell which is turgid. Okay. Normal turgid cell full of water. Normal turgid cell. And the next one, cell B, is a plasmolyzed cell. plasmolyzed cell, where you can see the cellular constants have constricted into the middle. From the solution, water or strong sugar solution, name the solution into which cell A was placed before viewing under the microscope. Cell A would obviously be placed in water so that it took up the water, and that's why it is turgid. And cell B, it was placed in strong sugar solution because it lost the water and became plasmolyzed. Under what conditions in the soil will the root hair cell resemble cell A and cell B? Under normal isotonic condition, under hypotonic condition, it will take up the water, and in the soil it will take up the water and it will turn turgid. And cell B, how does the root cell will behave? If the soil has a lot of solute concentration in it, like salt and sugar, then it will turn to cell B. So in this way, you have to frame your answers and write. Some are objective, some where you have to discuss it. Okay, next one, this is also a diagram, if you can understand, it's a diagram of plasmolysis. Diagram below represents plant cell being placed in a strong sugar solution. You have to study the diagram and answer the question. What is the state of the cell shown in this diagram? A plasmolyzed cell. So I can write it down as a plasmolyzed cell. Plasmolyzed cell. Very clear from the diagram itself. Name the structure that acts as a selectively permeable membrane. What is acting as a selectively permeable membrane in here? The cell membrane is acting as a selectively permeable membrane. Is it the cell membrane? Yes. Cell membrane or the plasma membrane. Name the parts numbered 1 to 4 in the diagram. So in the first part you can see it is the cell wall, which is there as it was. The cell membrane. Third one will be nucleus. Fourth one will be cytoplasm. Notice the fourth one, it is not cytoplasm. It is the empty content of the cell. It is not the cytoplasm, empty content. Cytoplasm got constricted, the protoplasm got constricted in the cell itself. Empty content of the cell after plasmolysis has happened. How can the above cell be brought back to its original condition? It can be brought back by putting it into a hypotonic solution where it can take up water. Mention the scientific term for the recovery of the cell. Obviously, it is deplasmolysis. Recovery of the cell by deplasmolysis. And we come to the last slide of today's discussion where we will talk about the short answer type questions. Now, short answer type can come as diffusion and osmosis, difference. We had already enlisted the differences. Remember to write the parameters and then divide the points. Then Turgor pressure, wall pressure. What is Turgor pressure? When the cell is turgid, it is inflicting the pressure towards the cell wall. All the cellular contents are inflicting the pressure, and as opposed to that, the cell wall is also inflicting pressure on the cellular organelles, so that is wall pressure. Plasmolysis, deplasmolysis, plasmolysis, just put up the definition and write. It is the constriction of the full cellular material when it is placed in a hypertonic solution. Deplasmolysis is the regaining of the original normal cellular structure where again the cellular contents are throughout dispersed. Okay, these are the differences. Now, reasons. You have to name the phenomenon and describe it basically. Resins swell up when they are kept in water. When their resins are kept in water, they imbibe water. Don't use absorb something. Use imbibe. Now that you know the term imbibe water. Grass is killed if salt is sprinkled on it. If salt is sprinkled on it, the grass cells will plasmolyze. So, they will lose water and they will be constricted. Don't write it. Write it as the grass cells will plasmolyze. But if the marks is more, if there is a 2 marks question, then you go into the plasmolysis process. You discuss the process of plasmolysis. Lastly, wilted leaves become crisp when placed in cold water for a while. When they are placed in cold water, these wilted leaves will take up the water. Again, imbibition will happen and they will be more, what they have said, they become crisp. Okay. Herbaceous plant growing in a well watered soil wilt on a hot day. Why do they wilt? Because the rate of transpiration on a hot day is more than the rate of water absorption, more than the rate of water absorption. That's why they wilt. Again, when you move into the evening part where transpiration stops because the sun rays are not there, so then the rate of absorption becomes more and the plants, they regain their original state, the leaves. So rate of absorption is less than rate of transpiration in on a hot day. That's why well watered plant, they wilt. So this is basically, these are the questions. So I hope you have enjoyed today's lesson. You go through the slides once more, and if you enjoyed, do not forget to like, share with your friends and subscribe to Learned Education. Thank you.